Today's topic is Chara. This is a very popular, well-known green algae, and we are going to study about its systematic position, its structure, and its reproductive peculiarities. Okay, so to begin with, we will first talk about the systematic position. So you can see it belongs to the kingdom Plantae, division Charophyta, class Charophyceae, order Charales, family Characeae, and genus Chara. This basic systematic position is something that you will have to keep in mind. This is very basic. As you all know, the hierarchy, kingdom, phylum, class, order, family, genus, species, that is your hierarchy. And this systematic position is something that will fetch you marks. So if systematic position is asked, you will have to write about these, right? So Chara is commonly known as stonewort because they are covered with calcium carbonate. Now this can be a short question. Why are these plants or algae known as stoneworts? They are known as stoneworts because of their structure. Because of their special structure, they are known as stoneworts, right? They have calcium carbonate on their surface. It is a multicellular thallus or plant body which is profusely branched. That means you can see this picture here. You can see the branches coming out from the nodes. It is a branch structure, or here also you can see it is a branch structure, and the plant body is divided into rhizoids and the main axis. Okay, now from here you will have to learn at least 2 to 3 names of common species that are found in India. The common species found in India include Chara brownie, Chara fragilis, and Chara zeylanica. So when you are learning these names, you will have to be very sure of their spellings. Since you are learning the scientific names, you will have to be very particular about underlining the names and also about knowing the correct spelling. So now we will briefly talk about its structure. First and foremost point that we should know is that they are macroscopic. There are some important features that you should know. First and foremost thing is that they are macroscopic. Macroscopic means what? You do not require a microscope to see. You can see these organisms with your naked eye. They are multicellular, have a thalloid plant body, and the structure is 20 to 30 centimeter in length. Okay, and it is differentiated into rhizoids and main axis. I had just now talked about that. Right, now here you see the cells are eukaryotic. Okay, that means what? They are advanced, they are not primitive. The nodal cells contain dense cytoplasm. You'll have to remember these points. A single nucleus and a few discoid or elliptical chloroplasts. Discoid means a disc-shaped or elliptical. Either disc-shaped or elliptical. Okay. Discoid or elliptical chloroplasts in the internodal cells, they have large central vacuole. Okay, the internodal cells have large central vacuole, hundreds of nuclei, and many discoid chloroplasts that lack pyrenoid. That means what? That is, pyrenoid is absent. You'll have to keep this in mind. Pyrenoid is absent. Pyrenoid is a big cross here, right? Now look at its structure. See, this is a diagrammatic representation of the plant body. As I told you, it is differentiated into rhizoids and the main axis. This is the Main axis, right? Rhizoid and main axis. Now see, the main axis is erect, long, branched, and differentiated into nodes and internodes. This means what? This is the main axis. See, this is the main axis. It has got branches from here. It has branches. So point from where branches are coming out is the node. And the space between the two nodes is the internode. Clear? See, this is the nodal region and this is the internodal region. The internode consists of single, much elongated or oblong cell. The internodal cells in some species may be surrounded by one cell thick layer called cortex, and such species are called corticated species. So, this cortication is not present in all available species, correct? See, it is in some species, not in all. Huh? In some species, they may be surrounded by one cell thick layer called cortex. And if that cortex is present, we call it a corticated or corticate species. It's a corticate species. The rhizoids are white. These are the rhizoids. Okay. Thread-like, multicellular, uniseriate and branch structures characterized by presence of oblique septa. This is an important point. The rhizoids are characterized by presence of oblique septum. The septum, okay, septa is plural of septum, right? The septa that is present is oblique in nature. Clear? Now see, the internode is composed of two types of cells. This is a little detailed study. You should know this because in case questions are coming, you should be in a position to answer it properly. Clear? So there are two types of cells. What are the two types of cells? Axial cells and cortical cells. The axial cells are central, elongated and cylindrical cells. So these 3 points, actually just take it as point number 1 and point number 2. It will be easier for you to remember, memorize it. And see, memorizing it or mugging it up is not the point. You should be in a position to understand it and recall it properly at the time of writing the answers. Clear? Now coming to cortical cells. These are elongated cells that are smaller in diameter compared to the axial cells. So the cortical cells are smaller compared to the axial cells and they are present as a layer on axial cells and originate from the node. So the cortical cells originate from the nodal region. Half of the cortical cells grow upward, referred to as ascending filaments, while the other half grow downward and are known as descending filaments. Okay, so you all know what is ascending, that which goes up, and descending is that which goes down. So the ascending filaments are filaments that are growing up and the descending ones are filaments that are going down. All right. Now see, one part of it has been shown here in the picture. The node is composed of two types of cells which are surrounded by 16 to 20 peripheral cells. 3 appendages arise from the node. How many? 3 appendages are seen to have been arising from the nodes. We will see each one of them carefully. Number 1. What is that? Axillary branches. The appendages are nothing but branches. Second is the primary lateral and third is the stipulode. Now we will see each one of them carefully. What are the 3 appendages? The first one is the axillary branches. Okay. These are branches of unlimited growth. Okay. These are branches of unlimited growth. That means they grow infinitely. There is no limitation to their growth. Axillary branches are nothing but branches of unlimited growth that originate from the older nodes, and they are also referred to as long laterals. These branches are also divided into nodes and internodes. Now see here in this picture. This is the shoot of unlimited growth, this one. This part, this shoot is the shoot of unlimited growth, and this shoot is the shoot of limited growth, which is known as the primary lateral. Now we will talk about primary lateral. These are what? These are branches of limited growth. So the axillary branches are branches of unlimited growth, and the primary laterals are branches of limited growth that originate in whorls. Whorl means what? Look here. How am I drawing? This is your shoot of unlimited growth. From here, from the nodal region, whorls are developing. These are your shoot of limited growth. Correct? Please don't write like this in your exam. Shoots of limited growth are also known as primary laterals that originate in whorls of 6 to 16 from the branches of unlimited growth. Okay? They are also known as branchlets. Here they were also known as what? Long laterals. Here they are also known as branchlets. Okay? Each branch is divided into 5 to 15 nodes and internodes. The node develops secondary laterals that are hair-like unicellular structures. Clear? The sex organs originate from the lower nodes of such branches. So this is a very important line. Where will you find the sex organs of Cara? From where every year you will see some or the other questions coming. You will find them from the lower nodes of, you will find them arising or originating from the lower nodes of such branches. Clear? See, we'll talk about this again later. This one will, this is just a bigger picture. Here you will see the internode, which I was telling you, the gap between two nodes. See, this is a nodal region. This is the node. And the gap between the two nodes, this part is your internode. And here you will see the globule, the nucleus, which are the sex organs. Correct? We will talk about this now. Coming to the third appendage. Third appendage is which one? The stipulodes. These are what? These are unicellular projections that originate from the lower nodes of the branches of limited growth. Okay? So where are they originating from? They are originating from the lower nodes of the branches of limited growth. That means what? Where? This one. Branches of limited growth here. Now see the picture here. If the number of stipulodes is equal to the number of branchlets, it is called unistipulate. If the stipulodes are double in number, it is referred to as bistipulate. Okay, these are some additional informations. If you remember, very good. So what is it? If this number of stipules are equal to the number of branchlets, okay, what are branchlets? Quickly recall. Branchlets are nothing but primary laterals. See, primary laterals are also known as branchlets. So if stipulodes, the number of stipulodes are equal to the number of primary branches or primary laterals, then what do we say? We call it unistipulate, and if the stipules are double in number, it is referred to as bistipulate. Clear? If the stipulodes are arranged in a single row, it is known as haplostephanous. However, if the stipulodes are arranged in two rows, it is called diplostephanous. Just see the picture. These are easy diagrams. You can draw it well in your exams also. See, you can see the nodes here. See from the, if this is the structure, if this is the structure, here you will have your primary laterals. From the, I'll just change the color. It will be easier for you to understand. From here, you will see smaller appendages. These red ones are your secondary laterals or stipulodes. These are your stipulodes. Clear? So these are the three appendages which we were talking about. So this is the basic vegetative structure or the structure of the plant body. You don't need to study too much about it. You have learnt about the axial cells, the cortical cells, all these things. Now we will talk about the vegetative reproduction that operates in para. Okay. Here you will see reproduction is of two types, vegetative reproduction as well as sexual reproduction. So both are quite elaborate. So you'll have to study both these in details and you can expect questions from both these parts. The question can come like this: write a brief note on the vegetative reproduction seen in Cara. Or it can ask you 2-mark questions like, what do you understand by the term this amylum star or secondary protonema, all these things. Okay. So you'll have to learn these very nicely. So let's start with bulbils. Bulbils are small, spherical or oval bodies that develop on the shoot or root nodes of Cara. Look at the picture. This is a stem bulbil and this is a root bulbil. A is stem bulbil. You can see the labeling here, and this is a root bulbil. Where do they arise and where do they grow? I mean, how do they look? They are oval bodies. See, structure is ovoid. It's not a perfect sphere. It's ovoid and they develop either on shoots or root nodes. Clear? They detach and germinate into new plants. Now, when these structures, see, when these structures, they detach from the mother plant, they start germinating into a new plant. Okay. Now, coming to amorphous bulbils. This is amorphous bulbil. This figure is your amorphous bulbil. Amorphous bulbils are aggregations of cells that develop at the node. Suppose this is your nodal region and from there you will see an aggregation of cells. Aggregation means what? A collection, group of cells. All right. This is known as amorphous bulbil. Now when these get detached from the mother plant, they germinate into a new plant. So in both these cases, you can see there is no union of gametes. Okay, there is no union of gametes seen here. Therefore, it is vegetative in nature. Now coming to amylum stars. Our next one is amylum star. What are they? Amylum stars are, now from the word star, you can understand that it has got some amount of similarity with the structure of stars. Okay. Now, if you see this picture here, the diagram here, you will see a star-like structure is visible. Right? Now see, amylum stars are another type of multicellular aggregation that has a star shape and is filled with amylum starch. That is why they are called amylum stars. Star because of the structure, and amylum has come from the name of the starch that is filled in these structures. They are filled with starch, which is amylum starch. Okay. And they are found in the basal region of the nodes. At the bottom of the nodes. Correct? They are found at the bottom of nodes. Last, we are left with secondary protonema. Secondary protonema are thread-like structures that are formed in the basal cells of rhizoids, and they have the potential to give rise to new plants. Now see, all these structures, if you see, bulbils, then amorphous bulbils, then amylum stars, then secondary protonema, all these things, they have the capacity, they have the potential to give rise to new plants without union of sex cells or gametes. Therefore, all these come under the category of vegetative reproduction. These secondary protonema, if you see, they look like structures like this, thread-like structures, they often look like this. Now, when they get detached, now they will give rise to new plants. They just look like this. Okay. All right. Now coming to sexual reproduction. Sexual reproduction in Chara. Clear? Now we will see what happens here. The mode of reproduction, this is something very important, and almost every year in every university, you will get some question or the other from this sexual reproduction of Chara. This is so favorite and important also. The mode of reproduction in Chara is oogamous type of reproduction. Okay, oogamous type of reproduction. And always remember, oogamous type of reproduction is very, very advanced. Oogamous reproduction is very advanced. Clear? Next, see, the male sex organ is spherical and ranges from yellow to red in color and is referred to as globule. So, in this picture, this is the male sex organ and this one is the female sex organ, right? The female sex organ is oval, greenish in color and is known as nucleus or oogonium. This one is your globule. See? This one is the globule and this structure is the nucleus. Okay? This is the globule because it is globular and the elongated structure is nucleus. The elongated structure is nucleus. Okay? The sex organs are born. This is an important line. Please note it. The sex organs are born on the branch of limited growth, mingled. Mingled means it remains entangled, mixed up with secondary laterals. Secondary laterals, where did you see? You have seen it here. The secondary laterals are nothing but stipulodes. Okay, you should be very familiar with terms. See, the female sex organ or the nucleus is situated singly above the globule. If you see the position, see the globule is here at the bottom and nucleus is situated little above the globule. The species of Chara can either be homothallic or heterothallic. Clear? Now quickly see. A mature globule is yellow to red in color, as I was just now telling you, and spherical in shape. Later, you can see the diagram here also. See, this part, what I'm ticking here is something that you should practice. This and this. You don't need to practice this part for exam. These are important. Especially these two, not even this. Especially these two. Okay. Each globule consists of outer curved plates called shield cells. These curved plates are called shield cells. Okay. and they are 8 in number. How many? They are 8 in number. Each shield cell gives rise to central rod-shaped structure called manubrium. This structure, this elongated part, this is the manubrium. This part is the manubrium. Okay. The distal end of the manubrium gives rise to globose-shaped structure called the primary capitula, which extends into the secondary capitula. Okay. Now see, if there is less marks in the question, don't write in much detail, write till here. But if it carries more marks, then you'll have to write all these things. Clear? The secondary capitulum, now, what is it doing? It is giving rise to antheridial filaments. The antheridial filaments are 2 to 4 in number. The antheridial filaments, these are the antheridia. Okay, here in this picture, you can see the antheridial filaments are 25 to 250 cells. 25 to 250 cells and are known as antheridium. Each antheridium can give rise to biflagellate uninucleate coiled antherozoid. This is the structure of antherozoid. See, you can see 2 flagella here. That is why it is called biflagellate, and it is uninucleate. It will have 1 nucleus and it is a coiled structure. See, you can see this coiling here. It's a coiled structure. All right. Now we will move on to the next slide. See, as I told you, globule is at the bottom, nucleus is at the top of it. Now coming to the nucleus. The nucleus of the female sex organ is oval. It's little elongated also. That is borne on the small stalk. It is made up of one central cell and a large egg at the top. Okay. The whole structure is covered by spirally twisted tube cells that are 5 in number. 5 in number, except at the top. Okay. And here, now see, this part is the corona. Corona means not the coronavirus, it is the crown. A group of 5 cells called corona cells are present at the top. It is a crown-like structure that is present at the top. Okay. Now coming to the fertilization process, what happens here? See, during fertilization, the spirally twisted tube cells separate to form 5 narrow slits. Okay. Only one antherozoid can enter the nucleus. Now you can see the nucleus here. Only one antherozoid can enter through the, enter the nucleus through the slit up to the site of the egg. The antherozoid and the egg fuse to form an oocyte. This thing you know, right? This is the male cell. This is the female cell. They will fuse to form an oospore, which is diploid in nature. Okay, this will give rise to germination in Chara. This oospore will give rise to germination in a to a new plant body. Clear? Now, if we quickly write about its life cycle, what happens here? See, suppose this is your plant. This is the Chara plant, right? Now, it will have 2 reproductive structures. One is the globule, the other is the nucleus. This will produce N cells, right? This N cell means what? N cell means what? Antherozoid. And this is ovum. Okay. Now this antherozoid and ovum fertilization will occur. This will give rise to oospore. which is 2n. This oospore will divide meiotically. Meiosis will occur as we were studying while doing the life cycle, if you remember. Meiosis means what? It will become N cells now. N will again become primary protonema. Primary protonema means a plant body. Primary protonema, right? This primary protonema will form the plant again. Here. So this is the basic life cycle of Tara. I hope you have understood the basic idea of it. Okay. Now this session will end soon. We will once again meet in our next class with our next topic, probably day after tomorrow. Now you just let me know in the group message whether this is correct for you or not. This timing is everything is correct for